The overall goal of this procedure is to isolate intact and enriched bacteria containing phagosomes.
This method can help us answer key questions in the field of immunology, such as changes in phagosome maturation and antigen processing when pathogenic microorganisms invade host cells. The main advantage of the technique is that it produces high-quality bacteria-containing phagosomes using simple steps and without the requirement of specialized equipment. Individuals new to the method would find it technically approachable in comparison to the known methodologies. We first had the idea to develop this method when we were studying the mechanisms by which intracellular pathogens hijack the phagosomes to their advantage. Demonstrating this procedure will be Saray Gutiérrez, a postdoc from my laboratory.
To start Salmonella typhimurium culture, use a bacterial loop to inoculate a single bacterial colony into 5 ml of brain-heart infusion broth. Then incubate the suspension in a 37°C incubator overnight while shaking. Transfer 1 ml of this bacterial suspension into 19 ml BHI broth in a conical flask on the following day. Incubate again in 37°C incubator while shaking. When the OD600 reaches 1, remove the flask from the incubator and transfer the culture into a 50 ml tube. Then centrifuge the tube at 5,400 × G at 4°C for 15 minutes.